So picture the wild crashing Atlantic waves right off the coast of Morocco. Beautiful, right? Well, hidden just beneath that churning water is a potential absolute game changer for modern oncology. Welcome to today's Explainer, where we're going on an incredible journey. We're traveling from the rocky marine botany of the Atlantic straight into the sterile, high-tech world of cutting-edge biomedical labs.
Here's the roadmap for our explainer today. We'll start with the ocean's hidden pharmacy, then look at the root of the weed, dive into the laboratory extraction, explore halting the cycle, do a quick scientific reality check, and finally look ahead to the horizon. Let's get into it.
Section 1. The Ocean's Hidden Pharmacy.
Meet our main subject, Gigartina Pistillata. It's this dark red, sometimes brownish-red seaweed with these elastic, kind of cartilaginous branches. You'll find it thriving right along the wild Atlantic coasts of Morocco and Portugal. Now, this is actually an edible seaweed, but researchers aren't exactly looking to toss it in a salad. They're fascinated by what gives it that elasticity. We're talking about complex structural carbohydrates called carrageenans.
Marine biology is really commanding the medical community's attention right now. As researchers point out, seaweeds are turning into one of the most attractive natural resources of compounds that can actually replace chemically synthesized ones. See, the marine environment is harsh. It forces organisms to develop incredibly unique chemical defenses just to survive. And we are just now beginning to harness those biological secrets for human medicine.
Section 2. The root of the reed.
To really grasp this research, we first need to understand the enemy we're dealing with: cancer stem cells, or CSCs. You see, within a tumor, not all cells are created equal. There's this highly resistant subpopulation of cells that have incredible self-renewal capacities. What's worse, they can enter a state of quiescence, which basically means they go dormant to hide from chemotherapy. These specific cells are the ones notorious for driving cancer relapse and metastasis.
Think of a tumor like a stubborn dandelion in your front yard. Traditional cancer cells are the leafy top of the weed. They make up the bulk of the mass. Traditional therapies act a lot like a lawnmower, right? They chop off the top, the tumor shrinks, and things look great for a while. But cancer stem cells, those are the root of the weed. If your treatment leaves the root completely intact, that weed is just going to grow back, and often it comes back stronger and more resistant. Scientists desperately need a way to pull out the root.
Section 3. The laboratory extraction.
Now, what is absolutely fascinating here is the sheer biological complexity of the seaweed itself. It turns out the plant's life stage drastically changes the chemical structure of the carrageenans it produces. So researchers separated the seaweed into two specific life phases. First, you have the female gametophyte stage, which yields a kappa/iota hybrid carrageenan. But then you have the tetrasporophyte stage, which yields a lambda/xi hybrid carrageenan. And this one is crucial because it has a much higher concentration of sulfate groups. Keep those sulfates in mind because they're going to be a really big deal.
Chronologically, the lab process looks a bit like this. First up, the team harvests the seaweed in Morocco and Portugal. Then, they use an alkali extraction process to pull out those pure carrageenans. Meanwhile, over in the lab, they aren't just growing cancer cells flat on a petri dish. They actually culture these 3D tumor spheres. Think of them as microscopic three-dimensional balls of cancer cells that are heavily enriched with those dangerous cancer stem cells we just talked about. Finally, they apply the seaweed extract directly to these 3D spheres.
And the results? Honestly, they were stunning. Less than 1 microgram per milliliter. This is the IC50 value, which is the concentration required to inhibit 50% of the biological function. In the world of pharmacology, a number this incredibly low indicates extreme potency. Both the female gametophyte and the tetrasporophyte extracts showed this massive potency against HT29-derived colorectal cancer stem cells in the lab. Basically, it was effectively destroying the root of the weed.
Section 4. Halting the cycle.
When comparing different colon cancer cell lines under the tetrasporophyte or T extract, the results get highly specific. Cellular viability absolutely plummeted in the SW620 and SW480 lines, dropping all the way below 20%. But interestingly, in the HCT16 line, viability stayed up around 75%. So why the huge difference? Well, it turns out SW620 and SW480 share specific genetic mutations, like TP53 and APC mutations, that the HCT160 line just doesn't have. This tells scientists something critical. This highly sulfated extract isn't just acting as a generic toxin, it's actually interacting with specific genetic pathways in those cancer cells.
So the researchers dug into the exact mechanism of action.
And it really boils down to cell cycle arrest. The carrageenans act as an anti-proliferative. Think of it like literally stepping on the brakes of the cell's internal engine. It stalls cellular division right at the G1 and G2/M phases, which are these critical checkpoints in the cell's growth cycle. By freezing the cancer stem cells in their tracks so they literally cannot divide and grow, the extracts drastically reduce the overall area of the 3D tumor spheres.
Section 5. Scientific Reality Check.
Okay, while these findings are super thrilling, we absolutely must ground them in reality. There is a massive difference between an early laboratory breakthrough and an established everyday medical therapy. Keep in mind, this was an in vitro study. That means it happened in plastic dishes and controlled lab environments, not inside the incredibly complex biology of a living human body. So no, this is not a proven human treatment, and nobody should be running out to eat raw seaweed expecting it to cure cancer. This research requires rigorous future clinical trials, and scientists still need to deeply evaluate any potential toxicity. As we always say, science is a marathon, not a sprint.
Section 6. The Horizon.
The main takeaway from all this is pretty clear. Combining traditional natural resources, like Moroccan Seamoss, with highly advanced biotechnology holds just incredible promise for the future of medicine. The ocean is this incredibly dense, biologically diverse environment. The very fact that a specific life phase of a single red seaweed produces a highly sulphated molecule capable of freezing cancer stem cells in their tracks, that is a massive testament to the hidden power of nature.
So I want to leave you with this final, somewhat provocative thought. If a humble red seaweed just tumbling around in the Atlantic surf holds the potential to fight off some of our most deeply resistant cellular enemies, what other life-saving secrets are hiding out there in our oceans? The depths are largely unexplored, and it's totally possible the next great medical breakthrough is just waiting in the waves. Thank you so much for joining me for this explainer. Keep learning and stay curious.